So we've completed discussing Mendel and our fundamental basis of genetics, your dominant and recessives, your codominant, your incomplete dominant, how we are able to actually apply the rules of probability to our various genes. So Mendel didn't even call them genes. We weren't really sure what they were. His work was lost, rediscovered in the early 1900s. And then we started trying to figure out, okay, what exactly is passing this information from parent to child? And we actually thought it was protein. Okay, so a couple of things. Number one, protein is considerably more complex than DNA. There are 20 amino acids compared to the 4 base pairs in DNA. Additionally, basically everything else in the body is done by protein. Okay, if you seriously, it's on the test and you're not quite sure what macromolecule group does it, pick protein. The odds are that you're probably right. Okay, so we had experimental testing using bacteria and bacteriophages, okay, which are viri that actually infect bacteria, show that DNA is the hereditary material in living organisms. So we're going to talk about Griffith's experiments, Avery's experiments, and Hershey-Chase experiments. So Griffith looked at transformation. So he studied the pneumococcal bacteria and mice. So pneumococcal is what causes pneumonia. So one of the first experiments that hinted, this was one of the first experiments that hinted that DNA was the genetic code material. So first of all, he noticed the bacteria was capable of being transformed, and we had 2 apparent strains. We had the R strain, which had no sugar coating, okay, and the polysaccharide, right? It's a sugar, it's a carbohydrate coating. The mice survived. In the S strain, there was a carbohydrate coat on the outside, and the mice actually died. So he did all these things. We inject live R into mice, and the mice survived. Okay, so remember, R is the type that was non-lethal. He injected live S into the mice. They died. This was what was expected. Okay, so we inject heat-killed S into the mice die. Okay, this is actually how the influenza vaccine, I'm pretty sure influenza is technically a heat-killed vaccine. So you take the virus, you heat the crap out of it and kill it, and so it denatures and it can't function anymore, but you still have the protein coat on the outside that our body then recognizes. We're good. We inject live R strains and heat-killed S strains into the mice, and they died. Okay. Whoa, what's going on here? My proteins have been denatured. What's happening? Okay. So Avery ended up verifying that the transforming material was DNA. And again, prior to Avery's experiment, it was believed to be a protein. So he purified chemicals from the heat-killed S cells to see which ones could transform live R cells into S cells. So DNA from S bacteria caused R bacteria to become transformed. Protein digesting enzymes, proteases, remember I told you ACE means enzyme, and RNA digesting enzymes, RNases, did not affect transformation. So the transforming substance was not a protein or an RNA. If I digest with DNase, however, transformation is inhibited. Okay, so the conclusion of this was that DNA actually is the hereditary material, but not everybody was convinced of this. Okay, this is just showing, so I have, here we go. I have protein, okay. Oh, we've still got R, RNA, I've still got R, DNA, woo. I have actually managed to transform it to S. Carbohydrate, I still have R. Lipids, I still have R. Okay? Only putting the DNA changed the heredity of the R strain. Now, Hershey and Chase did this really elegant experiment. So they used bacteriophage to show the genetic material was DNA and not protein. So a bacteriophage is a virus that attacks bacteria. It's this funny looking little spaceshipy thing. Okay? So what they did is we have radioactively labeled phosphorus, and we have radioactively labeled sulfur. So you're not going to find phosphorus in proteins, and you're not going to find sulfur in DNA. The radioactive phages were attached to E. coli, and then I centrifused the bacterial cells, causing them to pellet the bottom of the centrifuge. Okay. The bacteria that were infected with viruses with radioactive DNA were radioactive. The bacteria infected with viruses with radioactive proteins were not radioactive. Okay, so again, this lends evidence towards this whole that it's actually DNA that's doing the transformation. Okay, so right, I've got a phage, my radioactive protein is yellow. Okay, my radioactive DNA is green, and I do this and I test for radioactivity. The radioactivity here is in the liquid and not in the pellet, but down here it's in the pellet and not in the liquid. Structure of DNA. Chargaff analyzed the contents of DNA. He figured out that it contains 4 bases, right? Adenine, guanine, cytosine, and thymine, which we all know and love. He grouped them as purines, which are A and G, and pyrimidines, which are T and C. And purines and pyrimidines bind to one another. And then the amount of bases buried between species, but what he found out is that the percentage that you get of adenine is always equivalent to the percentage that you get of thymine, and the same goes for guanine and cytosine. So we now know that A binds with T and G binds with C, or as I like to point out, the sticky letters and the curvy letters. Watson and Crick and Franklin. Okay, so they used X-ray diffraction of a picture of DNA to determine its structure. The conclusion was that it was a double helix, okay, with an alternating sugar phosphate backbone and paired bases on the inside. Okay, we know that each pair is basically separated by approximately 0.34 nanometers. Each turn takes up 0.34 nanometers. Each turn of the helix takes up 3.4 nanometers, and that A, T, C, and G are bound together by hydrogen. I throw Franklin in here because bluntly, she didn't get any credit. Watson and Crick got all the credit for it. Rosalind Franklin, unfortunately, this was in a time when, women in science weren't so highly regarded, and she was bluntly sort of abrasive, but she sort of had to be given the climate that she worked in. Her picture, her X-ray diffraction picture, was actually given without her consent to Watson and Crick, and they published the paper first. And in fact, she was not given the Nobel Prize for it either, because you could not give it after death. And she actually, unfortunately, died of cancer, probably because she worked with a lot of X-rays. So DNA is in 2 strands called a double helix. The sides are made up of pentose sugar, deoxyribose, and it is bonded to phosphate, the PO4 group, by phosphodiester bonds. The center of the nitrogen bases are bonded together by weak hydrogen bonds. This makes sense. We want access to the inside, which we'll talk about when we talk about replication and transcription and translation. Okay. So deoxyribonucleic acid is the monomer of this are nucleotides. So this is what we see. We see our phosphate group, we see our sugar, and then we see our nitrogenous base. The 2 strands run anti-parallel. So one strand goes 5' to 3', the other goes 3' to 5'. 5' and 3' refer to the sugar. Sorry, not the sugar, the carbon position within the sugar. And then the strands are connected by hydrogen bonds between the base pairs, as we said. Adenine is double bond to thymine, and guanine and cytosine have 3 hydrogen bonds between them. And that is what it lovingly looks like.